Okay, let us start. Good afternoon, everyone. I hope you are all having a wonderful day. My name is Dr. Nilesh Ashokrav Karande, and I am an associate professor in the Department of Quality Assurance at the Institute of Pharmaceutical Education and Research, Wardha, Wardha. It's my pleasure to welcome you all to today's exciting seminar on the topic Neurometabolomics, a tool for the drug discovery. We are delighted to have such a distinguished group of the participants and the expert with us. Metabolomics is an incredibly dynamic and evolving field, offering profound insight into the intricate biochemical processes of the living organism. Today, we are here to explore the cutting edge advancement in metabolomics, its wide range of applications, and the immense potential in hold in the revolutionizing healthcare, agriculture, and environmental sciences. Throughout the course of the today's seminar, we will focus on the various aspects of the neurometabolomics and its critical role in drug discovery. We will be hearing from the renowned expert who will share their valuable knowledge and the perspective on how this field is shaping the future of research and the industry. I am truly honored to introduce our first speaker for the today's session, Dr. Dada Saheb Kokre sir, Professor of Pharmacology at the University Department of Pharmaceutical Sciences, RTM Nagpur University, Nagpur. Let's take a brief moment to go through Dr. Kokre's profile. Professor Dada Saheb M. Kokre, presently working in the Department of Pharmaceutical Sciences, Rashtrasanta Tukdoji Maharaj, Nagpur University, Nagpur. He also serves as the Director of the Research and the Development Cell at RTMNU Nagpur. This position allows him to foster collaboration and innovation across various research disciplines within the university by supporting interdisciplinary research and encouraging collaboration among faculty members, researchers, and the students. The R&D cell aims to drive scientific discoveries that can have a tangible impact on the society. He completed his B.Pharm from Shivaji University, Kolhapur, and M.Pharm in Pharmacology from Nagpur University, Nagpur. He has been awarded PhD in 2008 in the Faculty of Medicine, Pharmaceutical Sciences from RTM Nagpur University, Nagpur. Prestigious Raman Postdoctoral Research Fellowship has been awarded to him in the year 2015 by UGC India to pursue research activities at University of the Illinois at Chicago, USA. He has expertise in the field of the neuroscience, wherein he is keen to understand the neurobiology of different neuropsychiatric disorders, employing rodent animal paradigm, using different techniques like the immunohistochemistry, epigenetics, and the gene expression. He is instrumental to study the neural secretaries, brain loci, and the substrate protein, along with the neurotransmitter, especially neuropeptide, responsible for the development of the neuropsychiatric disorder and suggesting possible pharmacological targets to develop the drug for the treatment of different neurological disease conditions. His laboratory is equipped with the state-of-the-art facility for neuroscience research that includes operant conditioning, microdialysis, HPLC with electrochemical detectors, stereotoxic apparatus, cryosectioning, fluorescence microscopy, and neuropharmacological related several behavioral paradigms. Additionally, basic molecular research facility, that is electrophoresis, Western blot, and PCR, recently been added to his laboratory. He has more than 100 international publications, 2 authored books, 1 granted and 2 published Indian patents to his credit. He has successfully completed several research projects sanctioned by the federal funding agencies from India. and also doing consultancy services for pharmaceutical industries. His research group has published many innovative, cost-effective fabrications of the laboratory tools which are routinely used in the form of the consumables in the neuroscience research, such as the stereotaxic guide, cannula, dual cannula electrode assembly, and the bipolar electrodes for the rodent. He has been bestowed with the Young Pharmacy Teacher Award by Association of Pharmaceutical Teachers of the India. The RTM Nagpur University has honored him with the Best Scientist Award in the year 2017. Please join me in warmly welcoming Dr. Dadasaheb Kokre sir to today's seminar. He will be sharing his expertise and insight on neurometabolomics, a tool for the drug discovery. We are looking forward to a highly engaging and informative session. Now, without further ado, I invite Dr. Dadasaheb Kokre sir to share his expert talk on neurometabolomics, a tool for the drug discovery. Sir, the virtual stage is all yours. Please start the session.
Yeah. Thank you, Nilesh. Thank you, everybody. And good morning to all of you. First, I would like to thank the principal of IPER, Rajendra Ganjiwale sir, for giving me this opportunity. And Manisha Puranik ma'am, convener of this event, who had trust on me that, okay, I can give justice for this topic, although not that much expert in the neurometabolomics, but yes, of course, we are working in the neuroscience. Thanks to Nilesh, Abhiram, and all others for having a nice hospitality, virtual nice hospitality. So my topic is neurometabolomics, a tool for drug discovery. This is like little bit ignored, compartment of the metabolomics. Majorly metabolomics are there for the metabolic disorders, like it's a CVS or the cardiac or many other peripheral one, and much not been really studied for the neuropsychiatric disorders. So today my will be on biomarkers can be a very good tool in order to have a diagnosis of that neurological disorders, or that neurometabolomics we can use in the form of prognostic marker, or even that marker can be a very good substrate in order to develop the therapeutic molecules, so that they will have the modulation of that biomarker, and thereby some therapeutic effect will be offered by it. So it's a attempt that I am doing in order to give justice to the neurometabolomics. So first we'll begin with a drug discovery. So it's briefly, I'll just cover in 2-3 slides, and then I'll directly go to the main focus of our presentation, that is the neurometabolomics. In drug discovery, we are always trying to develop the therapeutics, and that therapeutics are such that so that it should not have a side effect, or then if at all it has, then there should be a minimum adversity should be associated with that molecule. And that molecule should be always based on some existing substrate or the target. Means like in brain, there are the many receptors or the protein components are available that can be used as an target, so that we can develop the molecule in the form of agonist or antagonist, so that they will have the modulation of that substrate and they will have a very good therapeutic effect. So we should always, should behind in order to know the new substrate. It is not like today what markers or the substrates are available, physiological or the brain substrates, that are the only substrate for which we have to engage them for the therapeutics or have some analysis of it. Not necessary. We'll have to always evolve with new substrate or the biomarkers or the neurometabolomics, so that we can have a better treatment strategies today or tomorrow. So main potential should be lies in the identifying the disease markers, the neurodegenerative diseases or the many psychological disorders. Now it has become the mainstay, or it has been at par to the diabetes or the CVS even. So in all those conditions, then our attempt should be to understand that, okay, there is a great potential in order to discover these markers, because they can be a very serious one in order to identify a therapeutic agents. So we have to do the thorough study with the pharmacokinetic, pharmacodynamics, and then we have to come up with a very good active pharmaceutical ingredients so that it will try to have the management of that biomarker that we are discovering in the form of neurometabolomics for several neurological disorders. Usually our process of drug developments that begin with the defining the medical needs, whether there is a medical need. So in the neurometabolomics, I would say like there is a tremendous need in order to develop the molecules. There is a need. Earlier it was not. Truly it was not. And even in our policy as on today also, this is not a very much prioritized items for the Indians. But yes, you will find many peoples are suffered with neurological disorders. So there is definitely a scope in order to develop or have a very good neurometabolomic front in order to find the such molecules. So in that category, we'll have to know the disease mechanism. So it is not like only markers are available. How markers will come unless you don't know the disease condition. So thoroughly we should know the disease, not only behaviorally, molecular one, or the biochemical, or the many other aspects. But I am saying like in all, very thorough understanding of that disease mechanism has to be done so that we can identify some lead into that one. So that we'll identify some physiological substrate, neurological substrate, or some molecule or the protein, so that it can become a drug identity, or that molecule can be manipulated by means of the API. And then of course we have to optimize all those lead components, and then it will go for the preclinical or the clinical investigation. So again, very briefly, these are the different phases of the clinical or the preclinical trials. So in the preclinics, we people work in the preclinics, and then we are trying to identify the different neurometabolomic markers, which can today may not be identified as a great one, but then yes, the much studies will be required in order to, or studies are warranted in order to develop them a very good biomarker for the neurological disorders. And then preclinical aspect, then we know the clinical aspects, and then ultimately that molecule, once it has been approved by the authorities, it will come to the marketing. So we will today, our main focus is on the studies in the animal models. So our main domain is to investigate, although it is not directly with the human subject, but if today we work on the animals and come up with some of the biomarkers, at least one biomarker will be a very good biomarker, and that will work into the human subjects, and it will be useful for the diagnosis, prognosis, or for the treatment purpose. So then doing neurometabolomic work in the animals, but at all it is being done from our side. So we understand the behavior of animal for that different diseases which are being induced into the animal by means of different agents or conditioning the animal, and then we study its behavior compartment. Then in that also we do the biochemical estimation. So we obtain the different biological fluids or the CSF even, and the cerebrospinal fluids. In that we search for the different neurometabolomics, which can be a very good marker. Also, we touch to the genetic part or the epigenetic part, and not only the biomarkers which are available into the fluids, but there can be a neurometabolomics which are available into a single cell even, and from that cell. So that will be the different part now, how to analyze it. But at least today we have to concentrate over whether it can be a very good biomarker in order to identify that disease condition. So for that, main like mRNA or even the genetic, the gene which is being very specific for the neurological disorders. And even we can do the histological studies, and through histological studies, the biomarkers or the neurometabolomics which are there in the histological tissue that can be identified, which can eventually be there into the different biological fluids, even the blood also, and through that we can do the analysis part of that one. So my focus is not like the analysis and direct work so that it will be useful as on today, but not on today it is useful, but then we have to do rigorous exercise in order to identify, uncover, unfold the different neurometabolomic. So tomorrow it can be a very good substrate or the neurometabolomic marker. Few definitions I want to highlight because I know the Shyam Masakapalli, the keynote speaker yesterday, he focused on the integrative metabolomics, or the Kanhaiya Kumar sir, he had deliberation on the bioprocesses for the antibiotic. So they must have given the introduction or the introductory part of the metabolomics, but I don't think that neurometabolomics has been covered by them. So before going into a very deep of the neurometabolomics, first we should know what is a metabolomic. So it is a overall scientific study wherein chemical processes in which metabolite is involved. So all the biochemical reactions, I would say, the biochemical reactions, there will be different, starting materials, and it's a biochemical reactions where enzymatic processes are being taking place, different intermediates are formed, and it can have even the end product. So in that during metabolism process, these metabolomics are there. So what are the metabolites? Metabolites are the very small molecules, which are the intermediates or sometimes even the end product of the metabolism process. So usually or ideally, it has been considered that the metabolome or the metabolomics should be the like end product, but it cannot be the only the end product of the biochemical reaction. It can be even an intermediate, which is a very stable one, means I'm saying biochemically stable and can have a longer duration to remain into the physiology, so that having that molecule or identifying that molecule as a marker can be a very good strategy. And of course, the metabolome are the small molecules that are there into a different biological samples, or even it can be there at a cellular processes. So this is just brief on the metabolomics. The different types of metabolomics in which like a targeted metabolomics. Targeted means, as on today, we know these are the metabolites or the neurometabolites. So it is like very well predefined, and we eventually certainly know the role of that one. Like it's a dopamine. So we know dopamine can be a very good marker for Parkinsonism as well as the schizophrenia condition. So in schizophrenia condition, the level of dopamine gets elevated in certain brain loci, or in Parkinsonism, we know that it has been totally reversed. It been goes down because it's a neurodegenerative disorder. So there are some known metabolites that are available as on today. So against that, if something can be developed, so it can be a targeted metabolite. So we can target to them. Second can be an untargeted metabolites, means there is a full scope in order to explore. It is not only the dopamine. Serotonin can be a marker for Parkinson, or can be a marker for psychosis, or any other monoamines, or the other small substances that are available in the brain in the form of, can be a neurotransmitter or the intermediate of some biological processes, but which are so far today, they are not known for their critical role, but yes, we have to explore them. So it becomes the untargeted metabolomics. Lipidomics, yes, there is a crucial role of lipid molecules, the intermediate lipid molecules in the biochemical processes, and that is majorly been covered for the neurometabolomics, lipid-like substances. There is floxomics, means here we have to determine the rate of metabolic reactions. At what rate this, because the different techniques are being used in which the radio-labeled substances or the intermediates will be developed. So how long they remain into that particular state and get converted into the another state or the another intermediate. So at what rate the clearance of that neurometabolomics happens. So that comes under the metabolomics, that is a floxomics. Pharmacometabolomics, yes, this is our field in order to check the response of that metabolite against the drug molecule. So when we give the different pharmacotherapeutic drugs, so when that agents are being given at different stage of that disease condition, so at that condition, what is the status of that biomarker that we have been identified for that particular disease condition? So that study biomarker along with the drug molecule and its influence on the biomolecule, that can be studied into the pharmacometabolomics. Of course, there is a food and nutritional metabolomics in which investigations are being done that how the dietary effects on the metabolic one or onto the health outcomes. There is even, I told you like in the last slide, there is a single cell metabolomics. So now the science has advanced, or the metabolomic stage, metabolomic studies or this domain has been advanced to a single cell metabolomics. It is not like it will be there in the biological fluid or into the tissue or into blah, blah. It will be in single cell. So that level means within that cell even, or like a malignant cell or the cell which is under degeneration, or the cell can be regenerating one. So in that particular cell, what are the, very, very unique things can be identified, which can become a very unique marker for that disease condition. So that can be even well explored. And yes, our today's focus is on the neurometabolomics. So usually metabolomics means people are identified in order to develop the different tools in order to measure the quantity of that markers. But we are being involved in order to identify the new markers for that disease condition. Because today's marker may not be the complete marker in the form of like which give a very good diagnosis or the prognosis of that disease condition. So we'll have to always search for the new markers, and that is an unending process. So these neural metabolites, in order to analyze and to explore them, and which are very much vital in neurodegenerative or the different psychiatric conditions. So again, introductory part, but not the metabolomics. It's a neurometabolomics. Neurometabolomics, so it is not like much old science. It is like only 10 years old, which has been, first time this term has been coined in, just 10 years back, which investigates for the metabolites in the neuron. Neurometabolomics, which is, they identify the metabolomics in the neuron itself. And from neuron, then it may go into the, the extracellular fluid, and then from it can go to the extracellular into the blood even. So in that way, there can be a glia, because the brain is not only the neurons, there is equally content, or the way of glia is also there. Yes, it is like we know it's a brain is nothing but it's a 73% water is there, and therefore cerebrospinal fluid or the brain tissues are there. So in all those components of the central nervous system, we have to identify the metabolites so that it becomes a neurometabolite. So that why these are much essential, because we have to uncover what the metabolic alterations that are being happen to the Alzheimer, Parkinson, this is very much glamorous disease condition, but it is not all. Even the behavioral or the psychological diseases, I don't know, miss, we can even become a capital for that one. After the diabetes or the cardiac, India can become a capital for the psychological disorder. There is a much rush, of the patient towards the psychiatrist or the psychologist. Then in that we have to have the analytical tools, means neurometabolomics to judge them, to rightly identify and to analyze them, that at what level they are there, in which form they are being available. So for that, different tools or that techniques for those tools we have to develop, like NMR, LCMS, HPLC, or even the immunohistochemistry. Many such tools we have to develop so that it will become a, it can be explored the neurotransmitter dynamics. It can be useful into even the synaptic processes also. So that level we are checking that, the pre or the post synaptic, and then the synaptic cleft. So in between how the interaction happens, and within that also that neurotransmitter, get degraded into a different intermediates, and that can be a neurometabolites, and through which it can be, these tools can be developed. And through that, then we'll have to ultimately develop the biomarker. So doing all these studies, we have to develop a biomarker so that we can have a early diagnosis of that disease conditions. So in that also we are working, like currently we are working on the schizophrenia, in which like the very, very early symptoms of schizophrenia. It is not like the positive or negative symptoms of schizophrenia, and by the time dopamine level increase, very earliest, like, there is a change in the things in which there is, it takes more time in order to get up to the different situations. So that is a reversal learning pattern. So that we are understanding into the animal. That can be a very earliest stage in order to trap that, yes, patient is suffering with a schizophrenia, so that eventually we can give some medication right at the beginning, so that it cannot become a clinical schizophrenic condition. So what animals are being used in preclinical investigation towards the development of the neurometabolomic markers. So there are n number of animals can be used in order to develop the neurometabolomics, right? It begins with the Drosophila to the zebrafish. Even the worms are a very good animal model in order to study the neurometabolomics. Monkey, of course, all rodents and the vertebrate, invertebrate, or up to the chimpanzee and all that. Even the non-human primates can be an animal in order to study the biomarkers into these animals so that we can check into the human subject and directly have the relevance to that one. And after all, we have to extrapolate the data from this animal and see its relevance to the human subject. It's a basic steps that are being used for the neurometabolomic studies is like we have to have a sample collection. Sample collection in the form of like suppose it's a neurometabolomic, and in that then it covers the central nervous system, so brain and the spinal cord. So when we focus to the brain, then it can be a neuronal cell, glia cell, or even we can go for a biological fluid that is nothing but the cerebrospinal fluid. So we have to have a sample collection. So how we'll have to approach for the sample collection? So if there is malignancy into the brain or the stroke subject, then we can even obtain a small, small tissue, and we can go for the histopathological investigation within that tissue, or even we can obtain the fluid, we can aspirate that particular material, or even the fluid in the form of CSF. So we have to have the sample collection. Then there is a sample preparation because the analytical tools are being at high throughput technique, and in that sample preparation is also a much studied part, and we have to do it very thoroughly. Then we have to go to the analysis, and then we have to have the data acquisition, and then see that what is the normal level and in a disease level, and even in the advanced disease conditions, in what way the level are being getting changed, and accordingly then we'll have to have identification of that disease condition, and then in against to that we have to give a treatment regimen. So I have been divided this neurometabolomic understanding into the three components. One component that is the histological component, which is clinically not been practiced much clinically, but then of course understanding some neurometabolomics at this stage, because it will give the understanding into the which brain area this particular neurotransmitter or the neuromarker, neurometabolomic is increased or decreased, so that we can have a very good understanding of that neurometabolomic marker into that disease condition. So this is preclinical study, so we can go at any level in order to explore it. Once we explore it, then in what way we have to translate that study into the human subject. That is a challenging work, but then at least we should understand into the animals one. So first will be the histological, and then we'll go for the biological fluid. So in that we actually obtain the biological fluid in the form of like CSF or the extracellular fluid, and in that extracellular fluid we determine the different neurometabolomics, and then it has become a very good marker for the disease conditions that we understand in our lab. And also at the third, the genetic and epigenetic level, we are targeting that the mRNA. mRNA is also the neurometabolomic, and that neurometabolomic, that mRNA or even the genetic structure, because when it get altered, it get transcript and translate into a very modified the structure, and accordingly the even like in Alzheimer, we know there is a tau protein or the beta amyloid and all that are being developed. So it is nothing but development from the genetic structure or the mRNA. So at that level, at the transcription level or at the post-transcription level, at the translation state, what changes are taking place to that protein so that it becomes a altogether different protein, and that is how it get accumulated, and then it becomes a origin for the development of the several neurodegenerative disorders. So in that way, we are our approach in order to understand it. So first part is a immunohistochemistry. So in that the neurometabolomic very specifically, very selectively, I am saying, because this is an antigen antibody reaction, and in that this neurometabolite has been identified such that very specific antibody is developed for that metabolite, and therefore very selectively we can identify that molecule. So for that method, first we have to anesthetize the animal, and we'll have to perfuse the animal, because there are some conditions through which we want that brain should be thoroughly perfused with the PBS, and then it has to be fixed so that there should not be any blood cells remain there, because in blood cells also there are many enzymes and the proteins which can mislead to the antibodies that we are using on it. So first is like we have to have the perfusion of the animal, and for the perfusion we are using the transcardial method, and in that transcardial method we expose the thoracic cage, and then we cut the ribs, and thereby we go to the cut the pericardium, and there is a heart, and in that it's an anesthetized animal, deeply anesthetized animal, and then at the left ventricle there is an introduction of the needle, so that needle will have the infusion of the PBS, and then that PBS, when it's a saline material, get enter into the circulatory system. It is a closed system, and then we give cut at the right atrium, and giving cut at the right atrium, then automatically there is a sudden oozing of a fluid that is takes place, and with pressure there is a we have the inlet of the PBS material, and that is coming out, so that it thoroughly been perfused the brain or the entire even the organism, and then it becomes totally free, blood free tissue that is available for the neurometabolomic studies. So in that, now we have to isolate the tissue, and isolation is a very skilled work. In that we have to see that brain should not be sabotaged, and then through in any way there should not be a damage, in order to have the isolation of that brain. Then you can have the approach, that is the anterior or even can be posterior one, and through that even we can have the dorsal or even the ventral approach, and through which we have to isolate the brain. Many such approaches are there, and then it is that how you have been taught in that way. This ultimate aim is for that brain should not be damaged, and it has to be isolated. So very skillfully we'll have to open up the skull, and then we'll have to just isolate the brain, and then brain we have to take and see that the totally perfused brain that is being shown at the middle. So that perfused brain then we'll have to take for the cryoprotection, and in that cryoprotection that we have to dip into the 10%, 20%, and 30% sucrose solution, so that it will be cryoprotected, because in next stage that brain we are using in order to have a section with the cryostat. So in this sectioning, we can have the two methods that we can use, the wax sectioning or even the cryosectioning. In cryosectioning, that brain has to be a cryoprotected, because it will be there into your environment, which is a minus 30 degrees centigrade. And in that environment, this is a closed environment into the cryostat. Then we have to cut the slices of the brain, because that slices we have to use for the immunohistochemistry. And then we have to have the application of antibody to that specific antigenic principle, which is a neurometabolomic principle that is available into the biological tissue, and that we have to identify. So for that, we are using this approach. Now, this is one such study in which the one neurometabolomic marker that we identified, that is called the GFAP, which is a glia marker, not like very specific, the neuronal marker. It is a glia marker, which is there into the astrocytes or all the glia cells. It is called the glial fibrillary acidic protein. It's a GFAP, abbreviated as a GFAP. So in one condition, that is a spinal cord injury. So we induce the spinal cord injury into the animal, and in that particular trauma, it's a traumatic condition that is being induced. Then in that traumatic condition, at what level there is an injury, that we have to understand. So for that, this can be a very good neurometabolomic marker, and that's GFAP expression is get elevated. So you will find that into the spinal cord tissue, the expression of that GFAP, which can be a very good marker. If its expression is going more, then we have to understand that, yes, what is the state and at what level there is a trauma that has been happened to the spinal cord. So then we tried our different agents, like in the form of CART, and then we had given the treatment of CART because it's a neurotropic or neuroprotective agents, and then how it has been reduced that marker expression, indicative of, yes, there can be a healing being developed by that particular molecule. So if healing has been advanced, and there is a very proper healing that is being taken place into that animal, so that has been explained by means of here. Like you will find there is a reduction in the expression. Even the CAR treated animal, we find like this neurometabolome or this neurometabolite has been reduced, because this has been expressed in that glia cells only when tissue is under traumatic condition, and then eventually it goes towards the development of the inflammation. So which is giving this sign of like, yes, there can be an inflammation, or inflammation at what stage, and then accordingly we have to see that the expression of that GFAP. So this is one neurometabolite that has been identified in our lab, which is there for the traumatic condition, and wherein not a neuronal but glia cell marker which has been studied and which has been checked into the cell, in which this marker gives us a very good identification regarding that, yes, how this can be expressed, and in the way how we have to control it, so that we have to have the different therapeutics to be given at the different stages, so that we can minimize the expression of this marker, which is an indication of the healing processes. This has been checked into the even Alzheimer condition. So Alzheimer condition, we know like there can be a very established one, which are the known one. It's like targeted neurometabolites, but there cannot be an end with only the targeted neurometabolites. We have to also develop the, we have to always been explore and develop the new markers, and then which can become a very good marker, so that which can give a right judgment of the disease condition for the diagnosis or for the treatment part. So in one such study that has been conducted in our lab, the GFAP that has been identified. So in one such study, we have identified for the mechanical trauma, but in such condition, it is for the Alzheimer-like disease condition. So in Alzheimer, we know there can be many symptoms or it's a neurodegenerative disorder. But in that condition also, we found like there is a significant enhancement into the expression of this neurometabolite, that is a GFAP. And its level is being increased in one animal model that is being developed. It's a streptozotocin induced that condition, which is being checked like, okay, whether behaviorally we have seen that there is a dementia-like condition into the animal, even the biochemical and by other means also we have confirmed it. But then whether there can be a very certain thing that is available into a brain tissue, so that which can become a neurometabolite or the marker, so that we can identify the disease condition. Then also the different enzymes can be a very good neurometabolites or the marker for the diagnosis purposes. So in one such research that is being conducted in the laboratory, in which we have checked the level of tyrosine hydroxylase, the TH, which is ultimately develop the monoamines. It can be even the dopamine or the catecholamines. So that tyrosine hydroxylase is being identified for the condition, and in that condition we had the fear extinction. It's a model. Means the first the fear has been developed into the animal, and then animal has become very frightening one, and then even how that fear is going to extinct, because we are putting that animal or condition for the another fear condition, so that from this one condition it will get extinction will happen, and it will go for how that reversal mechanism is taking place, that is being understood by means of the mesolimbic dopaminergic circuitry. But ultimate aim is what we should know that for certain such conditions, even because the fear is nothing but even the anxiety or the depression. So at large, we understand at the level of this like phobia or the anxiety condition. So then the level of this tyrosine hydroxylase, which is a enzymatic neurometabolite, and which can be studied for the conditions. Then the one neurometabolomic or the neurometabolite, which is a very good biomarker, BDNF, that is the brain-derived neurotrophic factor, BDNF, which is not there only into the tissue, it also then enters into the blood, and in that even the level of this BDNF can be checked. So, which is a marker for neuroplasticity. What is the neuroplasticity? That neuronal cells undergo some morphological changes, and thereby they alter their structures, and then automatically the neurotransmission, and thereby with all these conditions like Alzheimer or Parkinson, there is a much neuroplasticity happen. What plasticity? There is a the degeneration takes place. So plasticity is like very earliest stage that we have to check. So if there is a neuroplasticity being taking place into that particular subject, so that can be an indication that animal is going to be, or it will be suffer with that particular disease condition. Our aim that yes, in addiction, and in addiction that we understand by means of the ICSS, that is the intracranial self-stimulation. So we have in our lab the operant chamber, and in that we train the animal to press the lever, so that animal learn to press the lever, and that is how the reward develop into the animal, and then animal try repeatedly, and it becomes a reinforcement condition. So in that reward and reinforcement condition, we understood that yes, there is a neuroplasticity happens, because this is a model of addiction. So overall, how addiction is being developed. So initially, addiction development is taking place with a euphoric condition, rewarding condition, and then it becomes a tolerance, and then dependence, and then it becomes a totally addict one. So that earlier stages, when it becomes a rewarding to that subject, what are the changes that are being taking place? If at that time only, if we come to know there are some markers and being checked, so we can avoid that particular fellow to become an addict one. So for that, the neuroplastic marker, that is a BDNF, that has been checked, and we found that which has been increased, the BDNF expression is much expressed into a neuronal cell, and that has been identified by means of having a very selective antibody against that BDNF type, and then which has been treated and been bind specifically to that one, and which expressed by means of this immunoreactivity, and which has been even had the double level with the card cell, so that BDNF expression happens only in a particular cell, that is a card containing neuronal cells. So it is not only the neuroplasticity of non-specific neuron, no, then selectively these neurons containing these neurotransmitters are undergoing these changes, and thereby this very selectively we can go to that level of the addiction. In addiction, this study that has been conducted as a part of my PDF work into the University of Illinois, there we have studied the binge drinking, the binge pattern. Nowadays, it has also been taking place into the our community. Like people are drink only at the weekends, like Saturday, Sunday. It is called the binge drinking, excessive drinking only for a certain day. And then its impact on the adolescence. Means during adolescence, if such binge drinking has been happened, then how it make an influence on the adulthood. So in that, we identified some intracellular markers, the intracellular neurometabolites, which are available in the form of like CRAVE or the PCRAVE. So which are the cyclic AMP, which is a secondary messenger. And that secondary messenger develop or the give trigger to the signal transduction system, and in which it gives trigger to the CRAVE or the phosphorylated CRAVE. That can be an intracellular marker. So when we study a single cell neurometabolomics, so in that we can really identify such markers. Yes, we'll develop a very good non-invasive method in order to identify them. But yes, our aim in the animal study to explore it, whether they can be a very good marker. So yes, CRAVE and PCRAVE expression that has been really been reduced into a amygdala, and that to a central nucleus of amygdala, and that has been checked with reference to this morphometric analysis and identified, yes, this can become a marker. Cancer, yeah, cancer. I have given many examples, like it can be a skin, breast cancer, visceral, but yes, it's a brain tumor. So in brain tumor, not much, advancements has been happened, because the brain is little bit neglected one, or even the brain part. In order to develop the neurometabolites, we are always trying to develop the neurometabolomics that are available into the blood, so that we can withdraw the blood, and then in that we have to check that neurometabolites. But not necessary that all the neurometabolites that we are studying for the brain, which comes into the blood, not possible. They may go into the CSF even, or they may remain into the tissue, but can give a very good judgment in order to have a diagnosis, prognosis of that disease condition. So not like we have to always look at the easy way in order to identify them, but it can be even a invasive way. But at least in animal, we are understanding through invasion. But yes, of course, we have to work on that and develop the method in order to different method, like the PET, that scanning method, or in the positron, or even the MRI or the fMRI, the functional MRI is there, to which also we can judge the such different markers, and we can eventually develop a treatment for it. So like in the brain tissues, this Ki-67, which gives the condition like, okay, what is the level of that brain tumor, which is at the level of stage 1, stage 2, stage 3, in the form of malignancy, that can even be checked immunohistochemically. So with immunohistochemistry, because we know in malignant cell, there are certain proteins which are expressed only in that condition. So in the normal cell, we may not find them. So only in the malignant cell, or in the malignant cells also at particular stage, the different, it's a dynamic state. And in the dynamic stage, like at stage 1 of the malignancy, or this one, the special proteins are being developed intracellularly, or they can be even on the cell membrane. So in that, they can become a very good diagnostic tool in order to identify that malignancy, the break malignancy, diagnostic tool. Or even we can use them as a prognostic tool in order to check, the prediction, in order to judge them. Okay, this is in this condition, because during the course or in the progressive that disease condition, we require even the prognostic markers, and that can be developed by means of the immunohistochemistry. It can be a predictive even targeted therapy. We are very much prediction that yes, it is there, that this can be a marker, and it has been predicted that it is been there for this disease condition. So we can develop some, moieties or the agonist or the antagonist, so that it will try to, cover up this, will bury it, or it will totally be eradicated from that particular one. And yes, our aim is what we are doing the research, and we are trying to develop some novel cancer targets. So in that way, it is not only for the psychological or this one, but there can be a tumor condition that is there in a brain at a particular brain loci or the brain nucleus, and then it is manifest by means of some behavioral changes, and then we cannot only diagnose that, okay, this only a psychological disease condition. Behind that, even there can be a certain malignant cause can be also there, and that we have to investigate. Even the immunohistochemistry, we have to try for the cost effective. Means as on today, this tool is very expensive, because for every marker we have to develop that particular antibody, and this is the customized antibodies, because we know which marker we are been identifying, which neurometabolite is come across to us. So against that, we have to develop the antibody. It's a very costly affair. So we have to always try for the cost effectiveness. It's a very high sensitive technique, as I told you, because it's a even we can go for a monoclonal antibody, so that very specific epitope that can be developed from that particular protein, and it will be bind to that particular one. So it is a highly sensitive, special one. This is the much advantage. It's a special one, because you will develop a different method, and you will identify the markers into the blood or in any biological tissue, because it is very easy to withdraw and go for the analysis. But then I am saying the spatial resolution means like at that level of the brain. So within the brain, like it is in amygdala, hippocampus, or the hypothalamus, that marker expression is enhanced. So how can we go for the spatial resolution in order to identify that it has been developed into that particular area? So for that spatial resolution, we have to opt for the immunohistochemistry as a tool. We are using this for even the human subjects. We have to, take out that malignant tissue and send to the different centers in order to conduct the only immunohistochemistry. Even the Tata Research Center into the Mumbai or anywhere or any other organization, they have to undertake the immunohistochemistry in order to check that abnormal tissue or the malignant tissue, and then they identify that yes, the level of cancer. It's a multiple analysis. It is not means within one tissue we can go for the even 2, 3, 4, 5 markers, and for that not new tissue being required, or the new particular, or we have to again withdraw that biological fluid. Within that sample, particular tissue sample, we can go for a multiple neurometabolite markers in order to investigate the condition. Now, I have come to the second compartment of my webinar, and that compartment is we have to go for not only to the tissue level or all that, rather within the tissue, I am saying now we have to enter to a level that is a, logically the complete solution in the form of like genetic structure. I am saying we have to have the genetic or the epigenetic markers in the form of the proteins or the protein components, or the itself structure. The histone can be a one protein which is there into the genetic structure. And with the histone, like histone acetylation, histone deacetylation, when that reaction takes place to that particular one, then automatically the expression of that gene, it can be increased or decreased. So it happens with reference to the behavior. It is that not like the genetic changes, it will happen with only the heredity or no, which can also be influenced by environment or our behavior also. If we are always under too much stressful condition, then automatically the genetic expression also get changed because the environmental and behavioral impact is too much on that genetic expression. So then that genetic expression in the form of like mRNA, even mRNA is the altered structure of mRNA after post-transcription. That structure will liable to develop some abnormal proteins after translation. And then that protein can be an amyloid or even the tau protein for the Alzheimer or any disease condition. So ultimately, we have not to go and identify the markers that are available in the form of protein. We have to go beyond that and identify, because even the mRNA can be a very good neurometabolite, or it can be a metabolic marker in order to identify the disease condition. So not necessarily only the mRNA, I'm going before that, even the genetic structure, even the chromosome and the genetic expression, which will be altered by all those conditions that we have to check. So in such studies, some epigenetic and genetic markers that we have been identified, and there is one like TET1, like it is called the 1011, the transcription protein that is there on the DNA methylation. So the DNA genetic structure, it undergo methylation, and automatically, the rate of expression, it can be increase or the decrease. And then that rate increase or decrease, that we have to check by means of doing such studies. And for that, very selective antibodies that are being available in the form of like hydroxymethylation or the methylation, and thereby we can check the level of the gene which is responsible for. So like earlier, we identified BDNF as a protein, BDNF as a protein with immunohistochemistry, but here we are identifying BDNF as a gene. It's a genetic structure. And with that genetic structure, how that genetic structure changes, and then it is responsible for. So not like it has been trapped at the level of protein. So even before that also we can trap, and that can become a very good marker. Again, the mRNA, we targeted the mRNA, and in mRNA, like we use tool in the form of siRNA. These are like small interfering RNA. And that siRNA, what they do, this siRNA get bind to the, because we have developed such siRNA which are having complementary nucleotides, so that it will bind to that express mRNA. So that mRNA, before going that mRNA and create a trouble in the form of misleading and developing some abnormal protein, that mRNA can be trapped here. So this is it is not I'm identifying as a marker, I'm giving a solution. So here where we are trying to provide a solution in the form of the siRNA. So if there are some faulty mRNAs are being formed, and now they are going to develop a faulty protein, and because of that protein there is a neurological disorder, so we can trap. So we know that yes, this mRNA is a neurometabolomic marker, and yes, we know this has been altered one, and then it will cause something else, or even the normal, but it will excessively be expressed and will cause something disease condition. So in order to provide the therapeutic regimen, and therapeutic regimen is what? The siRNA is a therapeutic regimen, so that very small nucleotide sequence can go and bind to that mRNA, and it will not allow that mRNA to translate. Although it has been transcripted, but it will not translate, so that it will not develop that disease protein which is responsible for any particular neurological disorder. So such small siRNA has been developed, and in that one study, that is the study on the, again, it is on the addiction. Our mandate of the laboratory is we work on the addiction or the anger behavior or the learning and memory. So addiction is the major component, and that we understand by means of the operant chamber. So in the operant chamber, in a very specific area, that is the lateral hypothalamus, we understand that how we have to control, or the siRNAs are being given, then in what way it will silence the LSD1, which is a lysine specific histone demethylase, that particular mRNA, and which has been silenced by giving the siRNA against that, so that we can have the very good control over the expression, and accordingly we are not allowing that condition to form, which is a disease or prognosis condition. In another study, we use the siRNA that is for the CBP. CBP is nothing but a crepe binding protein. The crepe that we understand, that is a signal transduction, the cyclic AMP binding protein. So in that element binding protein, that crepe. So for that there is a CBP, and that siRNA is being developed. And when we had given that siRNA treatment, ultimately we found that there is a significant reduction into the liver pressure. Significant reduction in liver pressure. So the rewarding act or the reinforcing act has been reduced considerably, significantly been reduced when that siRNA treatment is being given to that animal. So indicating what? Because that mRNA is getting to develop the protein, and that protein is responsible in order to, build that mechanism of the reward and reinforcement, and that is being addiction one. So to reverse it, the siRNAs, which is very selective, been for CBP, been developed, and that is how it has been reduced. So this can be a very good neurometabolite marker. So that we have been studied many. So in that, one such is there. Now, the third and the last component of my webinar is the markers which are being very routinely being observed into the biological fluids. Usually biological fluids are being dominated in order to have the extraction of marker from that one. So these two tools, that is the genetic level, or by using the epigenetic or genetic studies, or even the histological studies, our main focus is to simply explore that this can be a neurometabolite. This is a neurometabolite we are exploring, and which is a very good marker in that disease condition, and which is again going with a very good prognosis one. Means progressively it is increase or decrease, and automatically we can develop a different strategy. Now, third is like very regular tool that is being used into the metabolomic research. Metabolomic research. So it will come into the neurometabolomic research also, that we'll have to take out the biological fluid, and then in that fluid we'll have to check the different determinants, which are the neurometabolites. So one such tool that we have established in our lab, that is a microdialysis tool. And in that microdialysis tool, that one, there are the two pumps. In one pump, we actually have the inlet, and that is how we, give the fluid to that particular biological nuclei. And then there is a one outlet through which also forcefully we take out that fluid. So forcefully there is a pump in and pump out that fluid, so that very continuously that fluid is going into that particular environment. So whatever extracellular metabolites that are being, neurometabolites that are being generated, and which will give the right identification of that disease condition at that level, means in situ at that particular disease condition, not anesthetizing animal into the normal habitat. We'll have to only simply subject to into the ball, which has been mentioned there. This is a one big ball in that we subject the animal into a very normal condition, not being even sedate that animal. It is a normal animal. From that home cage we are directly putting here, and then there will be two pumps through which there is an inlet and outlet, and that outlet is directly getting deposited and get collected into the fraction collector, and that fraction collector automatically for every after 20 minutes at the designated time that we have to enter there, it collects the diastylate, and then that diastylates get collected into that one Yes. Yeah. Now, can you see this slide? Hello.
Yes, sir. Yes, sir.
It is visible.
Hello.
Hello, sir. It is visible now.
Yeah. It's okay.
Yes, yes, sure.
Yeah. So I had given the introduction that how the microdialysis this works and the technique to which how we collect the distillate. So this is the one that equipment I have shown the picture of equipments and through which we analyze that when we inject that distillate into having the sample preparation injected into it one and then the different monoamines like the dopamine, serotonin, glutamate or the even the further metabolite of that one like dopamine, dopac or the 5-HT5-HIA that can be checked into through this method. So this has like newly been established like 3-4 years back in the lab. And here the very, very known neurometabolites, which are been there in the form of like dopamine or the dopac, which can be checked to that diastylate. So which is as I told you, like which can be a very conventional way in order to obtain the diastylate. And through that extracellular fluid, we are obtaining the diastylate and then it is being analyzed and then we can check the level and you will find here the level of the metabolites which are being there, like the CART, by giving the treatment of CART and this one, the dopamine level is being enhanced with reference to the CART because CART which stimulate the further rewarding conditions again and again. So our main intention is to look for that neurometabolite that is the dopamine or the dopac and to check that how the level of this neurometabolite change along with the condition of the reward and reinforcement, which is the reflection of addiction. The one more study in which the anger paradigm we have developed, and in the anger paradigm, because anger as such cannot be, because there are many methods available in order to check the aggression in animal, but not a single tool is available in order to read the emotion anger into the animal. So then with our mentor, Dr. Subedar, we have developed the anger-like animal model in which we fast the animal. And yes, there are the chambers and then we subject the animal. And yes, anger can be read by means of the different parameters that we have been said there, the biting behavior of animal. And it's a single animal that we are subjecting. So there is no aggression so that there will be a fighting between the animals. So then we read the anger behavior in animal and in anger, then what is the dominant factor or in what way this can be changed. So we observe that like the IHT level, which is getting reduced into that animal and the 5-HIA level, which is getting increased. The total turnover of 5-HT serotonin is being reduced because it has been known that in the anger condition, there is an increased level of dopamine and which drive that anger into the subject and the level of 5-HT is been lower. So it's an interplay between 5-HT and dopamine. So if 5-HT is dominated, then it becomes a normal condition and 5-HT goes down, then automatically dopamine level increase. And that is how by obtaining the metabolic fluid or the that distillate and we check the level of this one. So it's the last slide of the presentation and here I want to make there should be some take home message and in that I am saying like identifying marker is a continuous process. We cannot only rely on the available marker as on today. They are not 100% reliable marker which will convey the disease state, the progressive state, or even the therapeutic stage. Not at all. Anything cannot be a very complete one. So we have to always look for it because there will be a much variations with reference to the gender even, male, female, the race even, or the species, age, n number of factors are there which will alter the marker state. So therefore, we have to develop a very concrete marker which will not influence with all such influences and will remain a very stable one. And not only to identify, we have to also look for a very much cost-effective method so that which can be used for anyone, and through that it will become a very, very simple affair in order to diagnose the neurometabolites and through which we can even diagnose or do the prognosis or go for the treatment. So that is what I developed, as I express into my last point, which can be a perspective like whatever neurometabolites that we are been identifying, uncovering, which should be there at least in use for at least one, like for the diagnosis, not for diagnosis, then it can be a prognosis. Not these two, but it can be a therapeutic, so that we can develop a therapeutic against that particular marker, which can modulate that particular marker and which will give a therapeutic effect to that disease condition. Thank you, everybody.
Thank you very much, Dr. Dada Saib Kokre, sir, for such an insightful and thought-provoking session on neurometabolomics, a tool for the drug discovery. Your detailed explanations, real-world applications, and the research insights have certainly added great value to today's seminar. We are truly grateful for the time and efforts you took share your expertise with us. I am sure all the attendees have gained a deeper understanding of how neurometabolomics is transforming the landscape of drug discovery and neurological research. Now, we will take a few minutes for questions from the participants. Please. Any question from the audience? Sir, I have one question.
Yeah.
You explain details regarding that identifying metabolites for particular disease. So can we help some computational models or the AI models in support all these investigations in future or recent 5 years or something?
Yeah,
like
definitely, Nilesh, because in this era, we cannot keep us, isolate from this information technology or the AI one.
Yes.
We have to take help in order to accelerate the rate of identifying the markers or the neurometabolite or even the metabolite. Any markers, we have to take help of that one, or even that you mentioned, like the CAD-like tool, in order to develop that one. Because we know there are certain environment that is already existing. So in that environment, whatever new thing that you want to develop, the way it is not only for the identifying, but in that identified, if your molecule go and fit into it, so which can be a treatment protocol also, or even identification. There are many such protein subunits or in any form that metabolites are available, which has not yet been explored. So to know them and where it will be bind and in what way their structural alteration, and you want to use that computer assisted drug design. Of course, we can take help of AI and this the methods in which can sometimes be told like the alternative methods for the animal. So through that also, at least what will happen, we will increase the speed, and secondly, we will minimize the number of animals to be used for such studies. Definitely, it will have a dual advantage in order to use for the discovery of neurometabolite.
Any other question from audience side?
Thank you, Dada.
Thank you, sir. Thank you very much.
Okay. Now the session is going to conclude. Thank you once again for joining us today. We hope this session has sparked new ideas and encouraged further exploration in this exciting field of the metabolomics. We will now proceed to our next session. I request all participants to stay tuned as we continue with the more engaging talks ahead. Thank you. Thank you very much. The next session will start at 2 p.m.
Okay, Nilesh, shall I leave the meeting, right?
Yes, yes. Thank you very much, sir. Thank you so much.
Okay. Thank you. Thank you.